With this chapter, we begin Unit 3 of A&P 241 at Spokane Community College. In the muscular system unit, we're going to begin describing muscular system as a whole. This chapter is split into 3 parts. During the first part, I'm going to talk about the structure of muscular tissue. In part 2, we're going to talk about the physiology of muscle contraction. In part 3, we're going to talk about the muscle physiology as a whole. So when we talk about muscle tissue, we're going to talk about what are the functions of muscles, what is the structure of muscle tissue, and the different types of muscle tissue. Muscle is a fundamental characteristic of many things. It is essential to generating ATP or adenosine triphosphate. We have 3 different types of muscle tissue: skeletal muscles, cardiac muscles, and smooth muscles. This table describes the different types of muscle tissue. Now let's look at muscle tissue in terms of voluntary control. In terms of whether or not we can control the muscle at will, we have 2 different types of muscles: voluntary versus involuntary. Skeletal muscle is the only voluntary type of muscle, meaning we can control it at will. So here's skeletal muscles. So skeletal muscle is the muscle that covers our bones. So it covers our skeleton, basically, and it is voluntary. Cardiac muscle and smooth, aka visceral muscles, are involuntary, meaning we cannot control them at will. Now, cardiac muscle is heart muscle. Visceral muscles are smooth muscles that are involved elsewhere along the visceral organs. So in the GI tract, the uterus, the eye muscles, erector pili muscles in the integumentary system, blood vessels, those kind of muscles. Intestinal muscles, those are all visceral muscles. In terms of appearance, skeletal muscles and cardiac muscles are striated. Striated means striped appearance. So skeletal muscles and cardiac muscles have a striped appearance, whereas smooth muscles do not have a striped appearance. Another difference between skeletal and cardiac muscles is that skeletal muscles are multinucleated, versus cardiac muscles only have one central nucleus. So let's recap the different types of muscle tissues. We have skeletal muscles, cardiac muscles, and smooth muscles. Skeletal muscles are voluntary, cardiac and visceral muscles are involuntary. Skeletal muscles are striated with multinucleated. Cardiac muscles are striated with one nucleus. Visceral muscles do not have striations and only have one nucleus. Function-wise, skeletal muscles help with generating heat. They help us maintain posture and help with movement. Cardiac muscles help the heart beat and therefore help with pumping blood. Visceral muscles have various functions such as movement of food, movement of blood, and maintenance of blood pressure, movement of pupil size, uterine contractions, holding of fecal matter, erection of hair, and a lot of other functions as well. Other functions of muscles, including communication, movement of body parts, holding in contents of various organs, including the GI tract, movement of stomach contents, stability, posture maintenance, storing of substances. Sphincter muscles, so esophageal sphincters, anal sphincters, moving substances such as oocytes, babies, fecal matter, sperm, production of body heat. 85% of body heat is produced by the contraction and relaxation of skeletal muscles. Now, muscle cells have 4 distinct properties that any other type of cells do not. The first property is electrical excitability. Remember, epithelial tissues and connective tissues do not have these properties. So muscle tissues respond to electrical stimuli. So they respond to stimuli by nerve tissues. So they respond to chemical signals, aka action potential. We're going to start talking about what action potential is during this unit and the nervous system unit. So they respond. They cannot produce, but they can respond to electrical signals. The second property is the ability to contract, to shorten, as a response to electrical stimulus. The ability to extend or to stretch without being damaged, and the ability to come back to its normal size without being damaged. So the four distinct properties: electrical stimulus or electrical excitability, contractibility, extensibility, and elasticity are the four properties of muscle cells that make muscle cells unique. So here's the light micrograph of skeletal muscles, and you can see the stripes or the striations extremely clearly in this light micrograph. They are voluntary muscles and they are striated and they are attached to bones. Now, anytime we talk about skeletal muscle cells, they are referred to as muscle fibers, and they can be extremely long. So they're about 10 to 100 micrometers in diameter and about 30 centimeters long. So they can be extremely long cells and therefore are called muscle fibers. And these stripes come from different contractile proteins. So we're going to see what those contractile proteins are in just a minute. So each muscle fiber is composed of different types of connective tissue. So we're going to look at these different terms: fascia, epimysium. Anytime you look at the term mysium, it refers to a component of muscle. So muscle tissue is composed of fascia, which is a type of connective tissue, epimysium, perimysium, and endomysium, which are all types of connective tissue. You have certain nervous tissue involved in muscle tissue as well as vasculature for blood supply. So let's take a look right here. So this is bone. We're talking about skeletal muscle right here. This is tendon. Tendon is connective tissue that joins bone to muscle. Now what we're looking at over here is fascia. Fascia is connective tissue that kind of continues the tendon on to the muscle. So that right there is fascia. It is made up of connective tissue. Now epimysium. Epimysium is a layer. You're seeing it right here. Epimysium is a superficial layer made up of dense, irregular connective tissue, and it is continuous with the fascia. It's continuous with the fascia, and it superficially covers a muscle bundle. So epimysium is a superficial layer of dense, irregular connective tissue that covers a bundle of muscle, called the muscle belly, if you will. Now, underneath you have bundles and bundles of muscle fibers called fascicles. Fascicles are bundles of muscle fibers. Now, these muscle fiber bundles are covered with another layer of dense, irregular connective tissue, and that is called the perimysium. So know the difference between epimysium and perimysium. Epimysium is the superficial layer of dense, irregular connective tissue, continuous with the fascia, whereas the perimysium is the deeper layer of dense, irregular connective tissue that covers the fascicle. Now, the fascicle holds individual muscle fibers. So the dense, irregular, or the thin connective tissue that covers each individual muscle fiber. Each individual muscle fiber is called the endomysium. So take a look at this. You have the epimysium covering the fascia. You have the perimysium covering the fascicle. And you have the endomysium covering each and every muscle fiber. Now, each muscle fiber has little myofibrils. So next, we're going to look at what myofibrils are. Now, before we look at what myofibrils are, we got to look at where the muscle fibers are coming from. Muscle fibers come from the fusion of tiny stem cells. And the stem cells are called myoblasts. Anytime you look at the term blasts or genic cells, we're talking about stem cells. So myoblast stem cells are the stem cells for muscle fibers. And myoblast stem cells, along with cells called satellite cells, fuse to form muscle fibers. So there's not much to this. So you have myoblast stem cells that, along with satellite cells, fuse to form muscle fibers. So now let's take a look at the muscle fiber in much more detail. So here is a zoomed in version of a muscle fiber. Now let's look at what the term sarcolemma is. The term sarcolemma. So right here, the blue lines. So basically, sarcolemma is the plasma lemma, or the plasma membrane of the muscle fiber. So right here is the sarcolemma. This blue layer, this blue sheet, is supposed to be the plasma membrane, or the plasma lemma of the muscle fiber, and it is called here as the sarcolemma. Now, every once in a while, you'll see little tubules. So look at these blue tubules right here. I'm shading them green as I speak right now. So here's one, here's another one right here. So those are called T-tubules or transverse tubules. They're continuous with the plasma membrane or the sarcolemma. Now, these are tunnel-like foldings, and they are storage reservoirs for calcium ions. Take a note of this point. So the T-tubules are storage for calcium ions. So the plasma membrane of the muscle fiber is called sarcolemma, and they store calcium ions. Now you'll see here in brown a whole bunch of these striations, right? A whole bunch of these striations. That is the endoplasmic reticulum of the endoplasmic reticulum of the muscle fiber. That is called the sarcoplasmic reticulum, and that holds the calcium ions as well. Now the T-tubules are the gates. The sarcoplasmic reticulum is storing the calcium ions, and the T-tubule is the gate to releasing the calcium ions. So the sarcolemma, so you have the sarcolemma, you have the sarcoplasmic reticulum that is holding the calcium ions. So far we've talked about the sarcolemma and the sarcoplasmic reticulum. So everybody has to be crystal clear with what the sarcolemma is and what the sarcoplasmic reticulum is. Sarcolemma is the plasma membrane of the muscle fiber. Sarcoplasmic reticulum is the ER of the muscle fiber. What is the sarcoplasmic reticulum holding? It is holding the calcium ions. What is the gate to the calcium ions? The T-tubule is the gate of the calcium ions. Now the cytoplasm of the muscle fiber is called the sarcoplasm. You have the nucleus right here. Now remember, this is a skeletal muscle fiber, and hence it is multinucleated. You have another nucleus right here as well. Now we're going to look at what these striations are. So moving on to the next slide. So we talked about what sarcoplasm is. It's the cytoplasm of the muscle fiber. Now, it is filled with myofibrils. We're going to talk about what myofibrils are in just a little bit. It is also filled with something called glycogen. If you remember the first 3 chapters, you should know what glycogen is. It is a complex storage carbohydrate. It is a storage carbohydrate that is stored in muscle cells. Sarcoplasmic reticulum is the smooth ER of, and I already talked about this, and the T-tubules serve as gates and calcium reservoirs. Already talked about this. Make a note of this because when we go to the muscle contraction part of this chapter, which is part 2, we're going to talk about how calcium ions come into the picture in the muscle contraction cycle. The myofilaments. So these striations that you are seeing right here come from structural and contractile proteins. And those proteins are thick filaments. You can see that there are some filaments that look thick and some filaments that look thin. So they come from thick and thin filaments. We're going to go into a lot more detail about the thin and the thick filaments, and they are key to muscle contractions. You can also see a zigzag line that is kind of perpendicular to the thick and thin filaments. You can see the zigzag line right here. Now that is the Z line held together by Z discs. And between 2 zigzag lines is a functional unit of a muscle fiber. So between those 2 green zigzag lines is what is referred to as a sarcomere. And that is a functional contractile unit of a muscle fiber. So when we're talking about muscle contraction, we're going to see this as a unit of muscle contraction. So let's look further into one contractile unit. So to recap, I'm going to go over the muscle fiber structure one more time. We have the sarcoplasmic reticulum, which is the plasma membrane of a muscle fiber. This is the sarcoplasmic, this was the sarcolemma, which is the plasma membrane of the muscle fiber. This is the sarcoplasmic reticulum, which is the smooth ER of the muscle fiber. This is the nucleus. You have mitochondria. You have the T-tubules right here. You have the thick filaments and the thin filaments. You have the Z discs right here and the Z line right here as well. And this unit right here is the sarcomere. Now let's look at a sarcomere more closely, and this part can get a little bit confusing, so stay with me right here. Now, in a muscle fiber, the muscle contraction occurs due to the interaction between the thick and the thin filaments. Now, there are a lot of proteins that are coming into the picture right here. So each sarcomere exists between a Z line, which is right here in blue, and another Z line. Now each Z line is held together by Z discs. Here's a Z disc and here is a Z disc. Now, jutting inwards towards the center of the sarcomere from the Z discs are thin filaments. And thin filaments are made up of structural proteins called actin. So here you can closely see a Z disc. Here's a Z disc. Here's another Z disc. And another Z disc. So they're staggered in position, hence there's that zigzag appearance. And jutting inwards is a thin filament. So jutting inwards from each Z-disc is a thin filament. And it is a structural filament made up of the protein actin. Now, alternating between the thin filaments are thick filaments. And the thick filaments that you can see right here in the red are made up of the protein myosin. So thin filaments are made up of actin, thick filaments are made up of myosin. Now, right here in the center is a line called the M line. So when the muscle contracts, the thin filaments are sliding towards the M line. So there are other regulatory and structural proteins involved here that I'm going to talk about just in a moment. But these are the major two structural proteins. So when the muscle contracts, the thin filaments are going to slide towards the M or the middle line. Now there's a lot of other labeling going on over here. So let's talk about that. Now around the M line, there is a space. So take a look at this. The space on the thick filament from here to here. So the space on the thick filament around the M line is called the H for Heller, the last name of the scientist who studied this. It's called the H zone, H for Heller. So when the muscle contracts, the thin filaments are moving into the H zone towards the M line, right here. Now the A band, the entire width of the thick filament is referred to as the A band. A stands for anisometric. Anisometric. So that is the A band. And from the end of one thick filament to the next thick filament of the adjacent sarcomere is called the I band. I for isometric. So this is a little bit confusing. So write this down. Draw your own sarcomere. So draw a Z disc and a Z line. Draw another Z line. Make your own sarcomere. Then draw an M line. Draw your thin and thick filaments. And draw the H, the Heller zone. Draw an I band and an A band. Now each thick filaments are made up of myosin, thin filaments are made up of actin. So myosin, each myosin molecule looks like two golf clubs taken together and their stalks intertwined. Now take two golf clubs, have their stalks intertwined, and you have these two clubs loose like this. So that's what they look like. And each actin filament, now this is a fibrous protein. Each actin filament looks like a string of beads. Now actin has two regulatory proteins on top of it. Now this is something you need to understand how muscle contraction works. So this is what the thin filaments look like. The thick filaments look like the golf clubs. The thin filaments, these yellow beads right here, are the actin. This is the actin right here. Now the thin filament is covered by two regulatory proteins. The first one is called tropomyosin. The tropomyosin looks like a fiber. It looks like a rope. This brown rope right here. The tropomyosin, the actin, has these little brown dots on it. Can everyone see these little brown dots? Now the little brown dots are little binding sites for those golf clubs. However, you want those golf club binding sites to be covered. And this rope is going to cover those binding sites. This rope is covering up all those binding sites. So this is the tropomyosin. Tropomyosin is covering up all the myosin binding sites. So it's a regulatory protein. It's not a structural protein, it's a regulatory protein. You have another regulatory protein called troponin. This troponin is sitting on top of this tropomyosin. What is this troponin doing, however? It is binding calcium sites. It's preventing calcium on binding and therefore changing shape of the tropomyosin molecule. So you have the tropomyosin, which is doing A, and you have the troponin, which is doing B. So just think of it that way. So there's a lot of moving components here. So you have the golf clubs that need to attach to the beads. But they can't attach to the beads because you have the tropomyosin that is covering up the binding sites. Then you have troponin, which is covering up the calcium binding sites, therefore preventing the whole reaction from happening in the first place. Something has to trigger this reaction to happen. And it's a whole cascade process that we're going to see how this happens in part 2 of the lecture. So this is how it takes place. You see the thick filaments. This is what the thick filament looks like, a bunch of golf clubs. This is what the thick filament looks like. This is what the thin filament looks like. In this illustration, the thin filament actually looks thicker, but that's not what it looks like in reality. So the contractile proteins are actually the myosin and actin. The regulatory proteins are troponin and tropomyosin. So here's what happens. When muscle contraction has to begin, calcium has to bind to troponin, which is this blue thing right here. Then a shift takes place. When the shift takes place, this rope, which is covering the binding site, moves. When it moves, these brown dots, which are the myosin binding sites, get exposed. When the binding sites are exposed, these golf clubs, which are the myosin, attach to the myosin binding sites. And hence, muscle contraction can take place. And it's a complex, multi-step process. So we've seen the two major structural proteins, but there are some other structural proteins that come into the picture as well. The next structural protein is called titin, and it is the third most abundant structural protein after actin and myosin. Now, titin anchors the M line to the Z disc. So here goes, the M line to the Z disc, the spring right here. That spring is attached to the M line via titin. Now, alpha-actinin it forms the Z discs and anchors the titin. You have nebulin, which anchors the thin filaments to the Z discs, and you have dystrophin, which anchors the sarcomeres to the sarcolemma itself. So I already talked about what the striations are. The striations come from the appearance of the thin and the thick filaments. You already know what the Z discs are. The A band or the anisometric band, which is the dark band where the thick filament is located. You have the H band or the H zone, Heller zone, which is the center of the A band, the M line, the middle line, and the I band as well. So to recap, here is the M line. Around the M line is the H zone. This is the H zone. Here is the A band. Here is the I band. Here's the Z line. Here's another Z line. This whole thing is a sarcomere. Here you see these striations, which is the thin filament and the thick filament. You see the nuclei right here, the endomysium, which is fairly clear. And here is a transmission electron micrograph where you can see the M line extremely clearly. You can see the H zone as well. The A band is very clear. It appears dark because of the thick filament. The Z discs and the Z line is very clear, as is the I band and the entire sarcomere. So in the next part coming up, we will start off with the excitation, contraction, coupling, and look at the cycle of muscle contraction.